Yo, yo, yo. Today we're going to cover the scientific method. A lot of y'all already heard of the scientific method, but maybe you might not have heard of the details we're going to go over today. So maybe you've heard of this one. Ask a question, formulate a hypothesis, perform experiment, collect data, draw conclusions. Normally, a science teacher, middle school, junior high, even in high school, has gone over this scientific method, and it's just a simplified version of it. But in reality, it's a little more complicated than that. So a better version of the scientific method is starting off with observations. We move on into asking questions, and this leads to forming a hypothesis. And then we have a method of designing an experiment and then performing that controlled experiment. And from analyzing that data, we can make appropriate conclusions. And again, it's a cycle and it looks like a cycle. Boom! I bet you didn't realize that there's another slide that there could be something better. And in reality, the scientific process is truly nonlinear. In investigations, usually they have the same steps over and over again, and then with new information or new technology, they can conduct other or better their experiments. So the best version of the scientific method is definitely nonlinear, and this is what it looks like. The big middle concept is testing ideas. That's what science is, and it can lead to three other facets of the scientific method. So we have exploration and discovery. Exploring concepts or discovering new things may lead to testing new ideas, and from testing new ideas, we need community analysis and feedback without our fellow scientists to test if something is valid or not valid. We can not move forward or progress in science. Remember, science isn't the end-all answer. Oh, I use science in order to know that this is true or false. Again, everything in this world is uncertain. With better technology, we are better able to test these set foundation of ideas that we have. And then last but not least, the benefits and outcomes section. Again, if there are benefits, we can go back and test it to make sure that it is indeed not false. So going into the exploration and discovery part, there are parts within that section. So again, making observations, asking questions, these are part of exploration and discovery. Other scientists can find ideas from reading literature or research papers, and then finding inspiration in everyday life. So ways to move forward with these exploration and discovery is the introduction of new technology. Again, curiosity is a big one. Personal motivation, whether someone's trying to get their PhD, they have to go through this process. So very, very crucial. So an important part of exploration and discovery involves making observations, and that means no inferences. Inferences means to make a prediction or a conclusion about the things you saw. So again, observations only. Let's practice the difference between observation and inferences. So the first one, there was an axe next to the bloody dead body. So to me, this is a clear observation. We're just stating the position of the axe next to a dead body. The axe was the murder weapon. So this here is an inference. We don't know if the axe was used to kill. There was no autopsy yet. So that is an inference, not an observation. Next one. The axe at the crime scene was found 10 meters to the left of the body. So again, we're describing in detail the position of the axe, so 10 meters to the left. Last statement, the killer was left-handed and threw the axe to the left. So this here is an inference. Again, we don't know the killer's hand dominance. Okay, he could be right-handed, who knows? That is considered an inference. Using details, we draw a conclusion from that, and that should never happen in the exploration and discovery portion of the scientific method. So part of the discovery and exploration portion of the scientific method is ask a question. So in order to build a question, we could ask this question: what is previously known about the topic? And to help us build another question, we could use this question: what is it that still needs to be known about that topic? Good scientific questions lead to further discussion, leads to further investigation, needs to have measurable values, meaning you can measure something, lead to fact finding, as well as identifying independent and dependent variables. So on page 34 of your booklet, you can find this table here. And so now I'm going to give you guys a couple seconds to read over this, and you guys can fill it in on your own, but they cover the most important parts of an experiment: constants, control group, controlled variables, dependent variables, experimental group, hypothesis, independent variable, large sample size, qualitative data, quantitative data, repeated trials. So again, all of those are there for you to match, and you can match it on page 34 of your booklet. So is this a good scientific question? Is more fertilizer better for plants? If not, how can this question be improved? Right now, I want you guys to write on a piece of paper so you guys can later record your answer into the discussion board. How can this question be improved upon? Remember, for a good scientific question, we need to have independent, dependent variables and measurable values. In this question, can we measure everything from the independent variable as well as dependent variable? Okay. So testing ideas is the big section of the scientific method. So here we formulate the hypothesis. We have the expected results and observations, and the actual results and observations. And then that's in the data gathering. Interpreting data, again, we see if it supports a hypothesis, oppose the hypothesis. Does it inspire us to revise and create a new hypothesis? Does it inspire or revise assumptions? So the most important part of the testing ideas is the formation of a hypothesis. A hypothesis is basically a testable statement, or like I like to call it, a yes or no statement. It often contains a prediction. So an example of this would be sunlight increases the stem length of a plant. So either yes, sunlight does improve or lengthen the stems of the plants, or no, it does not improve or lengthen the stem of the plants. So we need to be able to gather data in order to support the hypothesis or oppose it. Or even maybe if we want to test it again, revise the hypothesis. So what's wrong with these hypotheses? Again, a hypothesis must take a stance. It must be a testable statement. The first one, chocolate may or may not cause pimples. In order to make it a correct hypothesis, or to make it a better one, we simply cross out the may or may not. It causes pimples. Now we can test it to see if it causes pimples or not. And then we can see if the data supports the hypothesis or oppose the hypothesis. Second one, I think salt in soil causes plants to grow slowly. So again, the I think portion is the incorrect part. You shouldn't use personal pronouns. Avoid including yourself inside a scientific hypothesis. So salt in soil causes plants to grow slowly. So there are ghosts affected by the color of the light. So again, it is a question. That is not a hypothesis. Remember, a hypothesis is a statement. Fourth, bacterial growth might be affected by temperature. So again, the might be, the wishy-washy. We don't want wishy-washy. We want definite answers. Bacterial growth are affected by the temperature or they're not affected by the temperature. You have to pick. Be decisive. And then last but not least, sparrows are happier birds than eagles. So happier is the incorrect portion of the hypothesis. Can we measure happiness? And I'll say no. You cannot measure happiness in birds. And it may be difficult, so this wouldn't be an ideal hypothesis. Again, maybe we might be specific in the type of neurochemicals within the brains of sparrows that could be in higher concentration than eagles. But happier, what determines happiness? Again, that is something that's not easily or clearly defined to be measured. So you might be asking, Mr. Tran, how do I write a hypothesis? So again, these are rules for writing a hypothesis. A hypothesis is written as a statement, again, not a question. The I think and I predict should not be found in your hypothesis at all. Do not include yourself in the hypothesis. And then a hypothesis is definitely testable. Again, remember the sparrow's happiness versus the eagle's happiness. You can't have a measurement of happiness. For a hypothesis to be properly supported through evidence or rejected, it must take a specific stand. No wishy-washy answers. You either go for it or you don't go for it. Again, it's a yes or a no. So here are some formats to help you guys with writing a hypothesis, an if-then statement. So if something about the independent variable, then something about the dependent variable. So that's a good generalized format of a hypothesis if you ever get stuck on one and you don't know how to start one. Number two, directly state the predicted relationship between the two variables. Number three, state the difference you expect to find between the two groups. So let's look at some examples. The first one, if a first-year student starts attending more lectures, then their exam scores will improve. So we see here attendance is the independent variable, and then exam scores is the dependent variable. Number two, the number of lectures attended by first-year students has a positive effect on their exam scores. So this is another way of writing the same thing. The attendance portion, number of lectures, is the independent variable. And then we want to see their exam scores, the dependent variable. Last but not least, first-year students who attended the most lectures will have the better exam scores than those who have attended few lectures. So same way of writing it, very valid. We still know the independent variable and dependent variable. We have a prediction between the two, and then there is a difference. So this is something that you may have heard of before, maybe not. The null hypothesis. This predicts no relationship between the variables. So independent, dependent variable, there's no relationship at all. There's no connection. And then you can also think of the null hypothesis as the devil's advocate hypothesis, because usually you're interested in showing some sort of relationship, but not always. So an easy way to show that there are some relationship between the variables is to say there's no relationship, testing that idea, and then if your data supports that there's no relationship, then valid. And if there is, then we can go further into the experimentation and revise the hypothesis to say one way or the other. So our example, plant growth rate is unaffected by the presence of cadmium in the soil. So again, it says that plant growth is not related to cadmium in the soil at all. So in the discussion board, develop a good hypothesis for the question you previously revised. So a reminder, is more fertilizer better for plants? Again, you should have created another question. If you haven't, take time to do now. And after you formulate your question, you may develop a good hypothesis. Remember, a testable statement saying yes or no. I will be looking in the discussion board, and Ms. Bossi will be looking in the discussion board to evaluate your hypothesis. So again, you can pause the video now to formulate your question and hypothesis. So, designing an experiment. Most experiments require a control group. This is the baseline or standard situation that does not receive the treatment. And this is the basis for comparison. One or more experimental groups is considered the groups that receive the treatment, different amounts of treatment. Other good points about experimental design is that it must include a random assignment to groups, control of extraneous variables, so no confounding variables must be found, and then manipulation of the independent variable, quantitative measurement of the outcome, and the comparison of participant groups. So going back to our example of fertilizers, so the amount of fertilizer would be the independent variable. So for our control group, we have no fertilizer, and let's see how the plants fare. And then varying amounts of fertilizers would be the experimental group. And again, we would want to be very detailed about the amounts of fertilizers in these groups. So the experimental variables is the independent variable, which is the variable being tested, the one that is being manipulated, the one you can control. The dependent variable is the variable that is being measured, very clear. And then confounding variables, other variables that must be defined and controlled so as not to be or to have confounding results. So types of data. Quantitative data, again, means numbers, easy. Quantitative, counting. Qualitative, quality. So these are non-numerical results. Examples would be color, smell, texture, presence of smoke, or sounds. The results can be subjective. So test groups should be of a large sample size, or the experiment should be performed several times to eliminate getting results by chance. And that's called anecdotal evidence. Okay, so we must retest, retest, retest in order to be confident in our results that it's just not by chance, but it's actually by the independent variable acting on the dependent variable. Common issues with experimental design. So the sample size could potentially be too small. When working in a biochemical lab like I did at Baylor College of Medicine, working with mice, the good sample size was considered to be 3 mice. So again, anything less than that would be considered not big enough to compare results or to be confident in your results. No repeated trials if I conducted that experiment only once. So that's bad. Uncontrolled variables. Again, things that we couldn't keep all constant could potentially have an impact on how we interpret results. No control group. So we didn't account for something to compare to. And then last but not least, we have one or more than one independent variable. So IV stands for independent variable. And that is bad. We don't know which independent variable is acting on that dependent variable to cause the results we see. So we only should have one independent variable. After the experiment, the results are analyzed and a conclusion is made. The hypothesis is either accepted or rejected based on evidence. Nothing is ever proved in science. It's just that we don't have the current technology to disprove a common knowledge thing. So now we're moving into the community analysis and feedback portion. So peer review is always important. We publish our results in publications or journals. And then we also discuss these results with colleagues to see if those results are plausible. So again, in the scientific community, all of that was listed there, and effects on researchers coming up with new ideas and questions from that discussion with the scientific community, and being able to start building a theory. The benefits and outcomes means that we can develop better technology in order to test new ideas, satisfy curiosity, build the knowledge that we have today, or solve everyday problems. So just as we have listed on the slide before, these are the benefits and outcomes of scientific research. Below this PowerPoint, you will find the cheat sheet to the scientific method. And on the next activity we're going to be doing today, we're going to be exploring the surface tension. How much water can a penny hold? So have at it, y'all. Have a good one.